The central nervous system is made up of neurons of the brain and the spinal cord. The nervous system consists of neurons or nerve cells which transmit information in the form of nerve impulses. Motor neurons transmit impulses from the CNS to muscles and glands that then cause a response. Whoa, what did you just watch? Well, let us explain. All neurons contain sodium Na and potassium K ions. Impulses occur as these important ions move in and out through the plasma membrane. Large negatively charged organic molecules such as proteins cannot leave the cell and are located in the cytoplasm. Outside the cell are the extracellular fluid, which contains more positively charged sodium ions and negatively charged chloride ions. These concentration differences are maintained by a specialized membrane protein called a sodium-potassium pump, which simultaneously pumps K plus into and Na plus out of the cell. When a neuron is stimulated, gated sodium channels in the membrane open and sodium ions from the outside flow in. They follow the electrochemical gradient to move into the cell. The neuron is now said to be depolarized. As charge is reversed in one area of the axon, local currents depolarize the next region so that the impulse spreads along the axon. An impulse that travels in this way is known as an action potential. Inside the axon becomes positively charged with respect to the outside as sodium ions enter. At this point, the sodium channels close. Gated potassium channels open and potassium ions begin to leave the axon, moving down their electrochemical gradient to reestablish the resting potential, a process known as repolarization. Because so many potassium ions start to move, the potential difference falls below the resting potential. Neurons have ion channels. The positive charge carried into the axon by Na positive at the spike initiation zone causes Na positive channels farther along the axon to open. More Na positive can then flow in and the process continues. The channels are triggered to open by the positive charge inside the axon, allowing more K positive to flow out of the cell and restore the negative resting potential.